as high as it was on the first day. But I'm here to break your spirits and tell you that whatever you have learned, you might have to unlearn quite a few points because there are challenges. There are challenges in every aspect of proteomics, and just like any technique, it's not foolproof. We had discussed it in briefs while we were discussing sample prep, while we were discussing about max quant analysis. But now I will discuss in even greater detail. But what are the specific challenges in proteomics as a whole? Specifically, what are the challenges that we get during data analysis? So first up is sample prep, and I told you, although it is not related to the instrument, it's always, remember, garbage in, garbage out. So an instrument is only good as the experimenter, right? A Ferrari in the hands of Michael Schumacher is incrementally better than perhaps in my hands. So what are the challenge? So there is a risk of experimental failure, and this failure might come because of incomplete cell lysis and protein solubilization. So the premise of proteomics is that we are solubilizing proteins, and these proteins are then further digested into peptides or proteolyzed into peptides, and these peptides then fly in the mass spec and we get the results. But what if your very first step fails? I think a few of you at least who are sitting in the audience would have felt that there are failures. Jinal would have felt that. Shambhu has felt that, and I know it firsthand. So sometimes proteins won't just solubilize, or specific proteins that you want to study won't lyse. So all this becomes a challenge. So since we are not getting into the detail, because every problem, every experiment will have its own unique challenges. So before you start an experiment, do read up about the biochemistry of the component that you want to. If you want to work on cell surface, it would be different. If you want to work on something else, maybe a nuclear fraction or a mitochondrial fraction, so these things might have challenges. Another big challenge is contamination during protein handling, during sample handling. You see, we are ourselves very rich in protein. We have keratin flying all around, and keratin is the most favorite contaminant. And almost every mass spec data, if you analyze it, you would find keratin. There is no way of avoiding keratin until and unless you are perhaps in a very clean room wearing a complete PPE overall, the ones that they use in BSL-3 facilities. Else, you would have keratin, but somehow people have found out methods to avoid keratin. But at the same time, you should also take precautions while you're preparing your samples. Suppose you are asked to fill up a tip box. Now, many people have this tendency to hold it by hand and put it in the tip box. The moment you hold a tip with your hand, you have introduced a new protein, keratin. And due to the homology, and since most of us work on animal cells, keratin will come up again and again. Detergent interference. We had talked about it when we were preparing that we need to avoid some detergents like NP40. We need to avoid it because what they do is they affect downstream analysis. They will intensively reduce, and how did they do that? They reduce the column performance, the LC column, the C18 column that you would be allowing your peptides to pass through. If detergent is present in that, you are more likelier to have a problem where the column performance would be compromised. Ionization is also getting compromised. So this entire premise that the peptides will fly, but peptides won't fly if they have detergents along with them. And even very small trace amounts of a detergent can suppress ionizations and signals. So you need to be very careful. So if you get into a greater detail, first reason that we discussed, that why is there an incomplete cell lysis and protein solubilization. So if you go to this first point, so we'll now get deeper into it. So protein variability. Now when you're talking of a cell lysate or a tissue lysate, usually we would like to believe it's something that is very uniform, right? But we all know cells are not uniform. Every protein has its own hydrophobicity, its own character biochemically, right? And they are again complex, but sometimes with a lipid, sometimes with a carbohydrate, sometimes with DNA. All these will impact how it is getting solubilized. And for that, you have to really check for the specialized methods available. Whenever you plan a proteomics experiment, go back to the literature and see for this specific type of protein that you want to interrogate. Suppose you want to work on plasma membranes, a membrane protein. What is a way of enriching the membrane fraction? What's the best way to get enrichment in the membrane fraction? Suppose you want to work on nuclear protein or mitochondrial protein. I myself, I had a project on mitochondrial proteome, and the biggest challenge was getting clean mitochondria, because you need specialized ultracentrifugation methods where you would get a very clean pellet of mitochondria. So you have to set up density gradient centrifugation with Perkol gradient so that you get a clean band of mitochondria, and then you have to again separate it, but contamination will always come, and that will impact your signal intensity, right? We know because there might be some abundant proteins that might again interfere. So you wanted mitochondrial fraction, but you took a bit of cytosolic fraction as a contaminant. For that experiment, cytosolic fraction, although a protein, is a contaminant. So these are the challenges. Another is, now if you think that you will increase the number of steps, you are increasing your loss. Always optimize the number of steps, especially for proteins if your sample availability is poor. You are working with a histopathologist on cancer who will give you FFP tissues, and those tissue, they will give you maybe a few shreds of paraffin sections, and you want to extract proteins from them. Of course you can do that, and of course your high resolution mass spec can work with as little as 500 nanograms. But fact of the matter is, to reach 500 nanograms, you might have to do 5 or 6 or 10 steps, and these 10 steps might end up with only 150 nanograms. So this becomes a big challenge. Whenever you think that your sample is very complex and you have to add more steps, remember that each step, none of the steps are 100% efficient. Efficiency is only around 50 to 60%. So you're losing out 50% of your sample with each step. Of course, it is time consuming and labor intensive. So how much time do you have? How many samples do you have to run? Suppose you are running a big clinical project where you have to go for 2,000 samples and you have 2 years to work with. That includes acquisition of samples, that includes preparation of samples, running the proteomics experiment, analyzing the data, coming up with a solid hypothesis. Can you afford to have a step that itself takes 7 days or 4 days? So there you have to make some compromises. Can we go for a step that can be scaled up? You might give up on a few proteins. So as I said, nothing is foolproof. The more you know a technique, the more you know how challenging it is, what are the pitfalls. And when you are designing an experiment, it's very important you know the do nots rather than the do's. Reproducibility. All these difficulties will pose another challenge, that is, how reproducible is your method? How reproducible is your method? The more the number of steps, the more the human intervention, the more errors that you will put. Even if you work with maybe 5% error, now calculate if you have 20 steps, how much error you have introduced. And this error, when you multiply with the fact that maybe there are 1,000 samples or even 100 samples, this error margin becomes so big that now your data is no longer reliable. You have compromised on data integrity, which is very important in science, that your results are reproducible. So this is a big challenge. So how do you deal with it? Of course, we have discussed this, that you can use Rapigest, Protease Max. So these are all companies, proprietary detergents that are compatible with the MS. Also, you have to check for phthalates. So when you are using a mass spec, so something that we always use is plasticware, right? Plasticwares leach compounds like phthalates, which will then go downstream and overwhelm the mass spec. So always make sure your plasticware is compatible with the mass spec. And how the one way of doing it is resourcing your plasticware from quality manufacturers. And if you know a lab who is doing mass spec, ask them what is the plasticware that you're using. So the problem is so vast that if you go for multi-center projects, they all source their plastic from one vendor with one single batch number. So even before the project starts, they know that I would require maybe 100,000 tips. And across 5 centers, I would need 500,000 tips over the next 3 years. They will purchase all those tips and ship it to the individual centers. Because even the best manufacturer in plastic, so we live in a globalized world, right? We do not know from which source who is coming, right? A simple thing, maybe this laptop, it would have components from all across the globe. The silicon is coming from somewhere, the plastic is coming from somewhere, the LED screen is coming from somewhere, the gold interfaces, that gold might have been mined in Australia, maybe in India, maybe in South Africa. You do not know. The battery components, they might be coming from different places. But over a batch, it is always same. So that's why you have to be very sure, especially when you are planning a big experiment, that all things come as a single batch. And especially if you are planning a multi-center experiment, again, all things should be coming from one batch and then distribute that plasticware and even chemicals to all those centers, so that you reduce the variability that is induced just by human intervention, or sometimes because lack of knowledge of the supply chain. Another, of course, is, as we discussed, immunoaffinity, abundant protein depletion, and well as contaminants. So we have already discussed that abundant proteins like serum, albumin, they are so much in amount that when you do a mass spectrometry analysis to understand what are those proteins that are impacting your biology, the only protein that will seemingly impact your biology is albumin. Others will simply be masked by the noise, because the dynamic range of an instrument is limited. And this limited dynamic range, just hold on. मैं आपका नंबर एक स्टूडेंट से शेयर कर रहा हूँ, आप वो बात कर लेंगे. हेलो मनमीत, इनसे बात. So yeah, have you started back? Right. One thing is immunoaffinity depletion that does is it may unintentionally remove other proteins. Now suppose your biomarker had a very strong binding affinity with albumin. Now you have used a column that by immunoaffinity it is removing albumin. Your biomarker is attached to albumin. What are the chances that your biomarker will still be in the mass spec? We had discussed protein-protein interaction, right? So we always assume that when we are doing an immunoaffinity of the top 14 proteins, we are removing those proteins. We are only removing those proteins. Sorry to break the truth, we are not removing these proteins. We are removing those proteins and all those proteins which have a strong binding affinity with the top 14 proteins. Again, one more big challenge. And this is completely unintentional. This is not our intention, right? When we design the experiment, we always think that we are removing the top 14 proteins. These are our abundant proteins and we want to make it okay for the dynamic range. But ultimately what we end up doing is we are also removing those interactors. Next, protein contaminants. We had discussed this. Keratins from skin and dust. So most of the dust you see is the skin we continuously shed. We are shedding skins like we are snakes on a daily basis. So we shed skin every day and that skin ends up being the dust particle floating around. So if you are not working in a clean room, however hard you try, you would introduce some contamination. Enzymes from lysis and electrophoresis steps. So we are always doing lots of enzymatic proteolysis, but at the same time when we add trypsin to a sample, there is no step where we deactivate trypsin. Of course we do that, but do we remove trypsin? No, we can't. So I think we did tell you during sample prep that do not add extra proteins just for the sake of it. You can add, but there are limits to it, because then you are adding again something like you're adding albumin to your depleted plasma. So enzymes like proteases from digestion steps, this becomes a contamination that we can't help. There is always a contamination. What happens? First of all, the mass spec will analyze peptides from contaminants instead of target proteins. Especially true for DDA methods. When we are doing data dependent acquisition, you see the more intense the signal, the mass spec will think, okay, this is an intense signal, let's delve deeper. And now your mass spec is wasting time on keratin, it's wasting time on trypsin. So reduces the number of useful spectra recorded. Next, there are mitigation strategies. So you can implement an exclusion list. So when you design your mass spec flow, which we have not gone into detail because I thought first you need to understand the basic. So there are ways where you can limit the exclusion, make an exclusion list based on keratin. You can make an exclusion list of all the peptides that keratin can make. Based on trypsin, you can make an exclusion list that do not look into any M by Z that comes up with these values. That you can do. But limitations will always exist when contaminants are inherent to the sample. Now suppose you're working on skin. Keratin is no longer the contaminant. Now keratin is the protein you want to look into. So this will put a huge challenge. Again, you have to design a new method. So it's very important before you start off an experiment that you understand the biology, you understand the limitations of the instrumentation. Another thing that we need is the conventional HPLC or the modern UHPLC fractionation. So always it is LC, MS. So the LC is that either it's an HPLC or in some cases it's ultra high pressure liquid LC. So what can we do? So what are the issues? Membrane protein solubility. So it requires specialization, special solubilizing agent like acid solvents, detergents and cheotrops. and these agents need to be diluted or removed from MS analysis. So membrane proteins pose a big challenge when we are talking of HPLC based fractionation. So we want special acidic solvents. You see, we are adding 0.1% TFA, right? We sometimes add cheotropes as well in the buffer. So these buffers try to maintain the solubility of the membrane proteins. Next, what you need to understand, the ionic strength and the buffer capacity. What is the buffering capacity of your HPLC buffer? What is the ionic strength of your buffer, right? These things you have to always keep in mind. Next, pH response affecting the retention time and peak shape. Last time when we were yesterday or day before yesterday, when we were showing you peaks, even Professor Sanjeeva showed you chromatograms. So pH response, the pH of your buffer, if it is altered, if your buffer somehow pH is not properly set, again you will get a huge lot of noise. So that is one another limitation. Issues with this SCX and HPLC separation. So you are using a column and the peptide elution is not okay in that column. Maybe the C18 material was not packed properly, or if the peptides, the way the peptides interact with the C18 material is getting influenced by the buffer and the elution is not proper. Especially important when you are going for unlabeled variation. So when you are doing labeling, you have challenges. When you are going for unlabeled proteomics, there this elution time, and in fact this imprecise peptide elution can be a challenge. Next, peptides appearing in multiple HPLC runs. Now what if you are not running enough blanks, and what happens is your blanks and sample, they start to look same. Suppose you are processing 100 samples and you are in a rush, or your column is compromised. So what happens? Few peptides, they so strongly bind with the column that they are eluted repeatedly. So there, suppose 100 peptides bind, peptide A, 100 peptides bind to the C18 column. When you're running your regular gradient, 25 will elute out, 75 will stay back. Now you run a blank, again 25 will elute, 50 are still staying blank, staying there. Now you run another sample, you add another 100. So now your column contains 150 peptides. This time maybe because they are more and 25% need to elute out, this time how much will it elute? Something around 37 peptides. Now this builds up over time, and by the time you are in your separate, you are into your 20th or 25th sample, the peptide is consistently eluting from the sample, even in blanks. So important thing to understand here is you can avoid it by running proper blanks and also observing if the blanks are actually looking like a blank. So blank should ideally be a very straight line on a chromatogram. Complex samples. So when these samples are eluting out, the singly charged peptides, they are the fractions which will elute first. They will elute first and they will dominate the entire mass spectrum, the chromatogram. The di and triprotonated peptides are not easily trapped in the column. So this again impacts your data. Then there are technical challenges in liquid chromatography. Difficulty adjusting equipment for multiple parallel columns. Usually in a lab you would have a high-leq column, maybe a C18 column. So multiple columns are present, especially those who have already worked in a mass spec environment would appreciate this fact. We don't work on single columns. But every time you change a column, due to lack of time, due to lack of precision, you don't change your instrument setting. So the same pressure is applied on a new column and on an old column. But the binding affinity of the C18 material after maybe 1000 runs have significantly changed. You should change, so the new column would be significantly fresh, it can handle a high pressure, whereas the old column, which is kind of choked up now, you have to run it on a lower pressure. But we don't do it, because if you lower the pressure, immediately the chromatogram elution time will vary. So you have to be very precise when setting these things up. This is again a big challenge. It needs lots of labor, it's lots of time. Column costs and stability. So columns are usually very costly and they are not very stable. So you always have to have good money in your facility so that you can keep on buying new columns, and also new columns might fail. And that we have observed with Professor Sanjeeva's lab when we was working with 3 mass specs. We almost had around 25 columns failing in a 8 or 9 months. 25 new columns. That entire batch had a manufacturing defect. And it's very hard to convince the vendor that you have done something wrong. Those who are working on antibodies, on Western blots, you would know. Suppose an antibody is not working. The first thing they will ask you, show us the blot. Then show us the protocol. Then they will give you an alternative protocol. Then you have to again work on it. Again you have to show us them the blot. So maybe 6 months will go in convincing the supplier that they were wrong in the first place. Then they will take, okay, we are setting up a replacement, and this will go through our finance section. So from the field scientist, it goes to the sales and finance. They are very hard to please people. They will again ask for a report. They will schedule an engineer visit. So for 1 month, for 1 year, you keep on convincing, and after 1 year, they will try to reach a truce. Okay, so we supplied 25 columns, they were wrong, we understand the issue, but we cannot issue 25 fresh columns. We will give you 10 instead. Now you will start bargaining. From 10, you will say, no, not 10, 20. Then say, okay, not 20, 12. You say, no, okay, not 12, 18. And finally you bargain at 8, something like 15 or 16. Then they will say, okay, I am now setting it to our consumables department. They will deal with it. Now the consumable department will ask you for all the proof that what went wrong, get a report from the field scientist, get a report from the finance, and effectively you have been pushed back by a couple of years. Else, if you have lots of money, you keep on doing the finding, apply for a fresh set of columns with a new vendor. You are going for a new vendor, you are not satisfied with your current vendor, they set in a new column, now you have to set in a new method. Right? So you have lots of cars, you are driving one model, you are shifting to the other model. At least initially for the first 1 week, you will be very skeptical how this car will drive. How does it accelerate? How does it brake? Try, if you have a bike, try riding your friend's bike. Its brake response, its throttle response will be significantly different from yours. Same happens with instrumentation. So what we need to do is need for advanced column technology and bringing every system at par, right? So that columns become instrument agnostic. When a column becomes instrument agnostic means whatever instrument you use, all the companies are making a similar type of column with similar type of settings. Very hard in a market where everybody wants patents. Even our universities wants patents. Your CV should have a few patents. So whenever you patent something, you're hiding something. And the moment you are hiding something, it's no longer open source. and something which is not open source cannot be effectively copied. Next, the trade-offs between resolution and speed. So anything that is fast, you will agree. So if you have to cook a good food, it will take some time. But if you have to have something very fast food, it can do it very fast, right? You are compromising on the taste because now you have to eat something. So you can have 2-minute noodles, but if you have to cook a good wholesome meal, it will take you around a couple of hours. So if you have to compromise on speed, first then you have to compromise on your health. You know noodles is not good for you, but at the same time it's saving you time. So you decide, okay, let's save time today. That's what pharmaceutical and clinical labs do. They need to sacrifice resolution, because the moment you increase the resolution of a mass spec, you have to run a longer gradient, right? So a gradient that we use in research labs can be as long as 120 minutes. A pharmaceutical lab does not have time, or they do not desire to change the speed. So what they need to do is, they will essentially sacrifice resolution, run shorter gradients, and somehow get the data. So whereas fundamental research, like suppose you're working on a pathway and you need to have a much greater resolution, so you have to have a longer dynamic range, you need slow elution, so you will run a 2-hour gradient, and that gradient will give you much greater proteome coverage, because now you want to analyze a very complex mixture, you want to do some drug development, you want to identify a novel biomarker. So this is so I mean, you can say this is one of the issues with any experimental design. Even if you want to have to scan for a biomarker, once you have established a biomarker, you want to scan, you will just scan for that specific M/Z ratio, even if it is a orbit trap or a triple quad, right? You will perhaps run a PRM. PRMs are much faster. You can run it in 30 minutes. So thing is, we need to continuously improve upon these HPLC and UHPLC system. We can have a better resolution in mass spec, and we are doing it. Like as I said, 2 years back, people were getting around 800 or 900 proteins in plasma using DDA. Then you move to DIA, you get 2000 proteins. You move to a higher spec timsTOF, or maybe an orbit trap Astral, Thermo Fisher Astral, then you can push the number of proteins identified something around 4,000, 5,000, again depends on the method. And this is really required if you want to elucidate the biological mechanisms and you want to know the real story behind the disease development. So we had already discussed few things, but again I think it's good to understand what are the challenges in MS instrumentation. So mass spec is a very high cost, high maintenance instrument. The reason you won't find a mass spec in an average lab. You might find a real-time PCR, you might find a spectrophotometer, all these things, but anyone from a lab which has a mass spec of its own? Anyone in this room? Perhaps no. Anyone who has it in the institutional central facility? 1, 2, 3, 4, 5, 6, 7. So that's the thing. Most of these instruments are capital intensive instrument, costing crores of rupees, and they are very delicate as well. As I said, a small change in column might put things off. A small change in pressure, things will go off. So most labs require mass spec labs. A good high performing mass spec lab are run under 24-hour supervision. All the time somebody is keeping an eye. You don't need to be in that mass spec room. You can use softwares like AnyDesk to be at home, or while you are traveling you can share the screen and you can have remote access, but at the same time you have to look at it. If the pressure is falling, if the pressure is suddenly very high, if the mass spec has stopped giving you readouts, suddenly you're seeing even your samples are giving a straight line. All these things, and of course troubleshooting and maintenance, extremely difficult. So we had 3 mass specs in our lab at IIT Bombay when I was doing my postdoc. The vendor was 2.5 kilometers away. The guys who sold it, their India head office was 2.5 kilometers away. And guess how much time they used to take to come to the office if there was a maintenance? How long would you take to travel 2.5 kilometers? 4 hours. They usually took 4 weeks. So you have to raise your complaint. Somebody will tell, okay, we are looking at, give us access. Are you sure this is the problem? Just like with any customer service. Once they have sold you an instrument, their customer service takes a back foot. Then their maintenance division wants to sell you more spare parts. इसमें ये लगेगा, उसमें वो लगेगा. So that's the biggest challenge. Data complexity. So one more thing that is having instrumentation issues is extensive protein information can lead to false negatives and positives. As I said, we only assume that this is the protein based on its proteome, M by Z signature. So we match the signature from theoretical spectra with the practical spectra. But thing is, this can lead to false negatives and positives. We'll come to discuss why. Unassigned spectra complicate the data analysis. So as I said, if you have too much of contaminants and all these kinds of unassigned spectra, the instrument will anyway try to make sense of it, because until unless you tell it not to look into something, instrument mass spec is actually somebody who will look into everything, right? A simple thing is you can take up the whole proteome data of the blood, right? Now in today's world we know that blood also contains some microbes, right? So blood contains healthy microbes, right? Maybe our gut contains microbes and they leak some proteins. Now do you think when the mass spec is looking into the mass spectrometry, when they are looking into the data, do you think they're only looking at human specific information? No, they are peptides. So any peptide, even those peptides that came from your hand while you are eating chicken burger and then you forgot to wash off your hands and somehow that chicken burger fell in your sample prep, a part of it, they will also analyze chicken proteome. So this takes and complicates. Again, instrument limitation. No single instrument can perform all experimental types. So if you want to do a really good quantitative proteomics, targeted proteomics, a triple quad is a better instrument, a triple quadrupole. If you want to do discovery, a hybrid instrument like Q-Exactive from Thermo Fisher, it's a good experiment, but there is no thing such as I have one experiment, I have one mass spec, and now I can do all kinds of proteomics. You can only do bottoms-up proteomics. For a top-down proteomics approach, you would need a different kind of mass spec, right? There are multiple types of mass spec. Since it was only focused on proteomics, we didn't go into those details, but you can look up that there are so many different types of mass spec, and each mass spec has its own strengths and flaws. Dynamic range, we have already told, they have a very short dynamic range, and that dynamic range might again pose a challenge. When we are talking of biomarker discovery, the biggest problem is the dynamic range and high complexity in biological samples. Biological samples by nature are very complex samples. You have lots of proteins. Take up a cell, think of the proteins in the plasma membrane. Are they hydrophobic or hydrophilic? They are both. The transmembrane protein the cytosolic domain and the extracellular domain, they will be hydrophilic, but the transmembrane domain will be hydrophobic. So one protein, you don't know whether it is hydrophilic or hydrophobic. It can be hydrophilic and hydrophobic based on the domain. Now, these kinds of information can make the sample complex over and over again. And these complexities add up, and it becomes a big challenge for the mass spec to go deeper into it. Low abundance of disease-specific biomarkers. Usually, disease-specific biomarkers are usually very low in abundance. They are not very high because they are not constitutively expressed. So when you're taking a blood sample from somebody, this blood is like the sewage system of our body, right? It goes through every cell, picks up small proteins that are leaking into, and then it keeps on transporting those signaling molecules. So if I have a small cut in my hand here, even if it's a very deep cut, all those inflammatory molecules and proteins, what are the chances that if I have a blood draw here, it will be coming? The chances are very low. So I've had around 6 liters of blood, maybe only 5 or 6 ml is actually having that information. Now, moment it spreads down, it comes down thousands of times less in concentration. Now it becomes a challenge for the instrument because, again, we had discussed that dynamic range is an issue. It will not pick things up. Instrument inefficiency, of course, we discussed that no instrument can fix, there is no one size fits all. Qualitative and quantitative proteome characterizations, again, remains a challenge. Now let's move on to what are the challenges on mass spectrometry data. The ones that, so after all these challenges, you get a data, right? You get a raw file that you can set up on MaxQuant for an analysis, or NMS Tragger, or Proteome Discoverer, depending on, or Spectronaut, depending on your instrument type and depending upon your lab funding. So for your rich lab, you will use Proteome Discoverer or Spectronaut. You are from a poor lab, you would perhaps use MaxQuant and MS Tragger. Not true, but that's how things work. Some people have the money to subscribe, and those subscriptions are really costly. I think around 8 lakhs per year for Proteome Discoverer. One user. You can have only one computer in the lab running Proteome Discoverer. And suppose there are, and sometimes even rich labs can't afford it for the very simple reason that they are having 5 or 10 mass specs, and it's a big team of 50 people working. So you cannot take 50 individual subscription. And the group subscription is also inordinately high. And if something free and open source can solve the problem, why should I spend money? Because you see, scientists are very challenged when it comes to money. And nowadays, there was a time when you used to get money and you used to publish free. Now you have to even to publish, you have to pay money. So this entire industry is about paying others money. So once the data is generated, the biggest challenge is peptide identification. Why? We have already discussed. We identify peptides based on the M by Z data of known proteins and theoretical fragments. Two things you have to remember: known proteins and theoretical protein fragments. That means a mass spec and a software like MaxQuant is limited by two things. Number one, it can only look at proteins which are already known. The reason I'm telling you this is we have a lot of unknown proteins, right? Professor Sanjeeva in the very first lecture told us that a significant portion of the proteome is unknown. So we are working on an assumption that whatever we are doing, matching them by Z data of known proteins and theoretical fragments, so this is an assumption that my sample based on my database can contain only about 8,000 proteins. We are assuming this. It might contain 80,000. Is there any way to know? No. It assumes all protein coding genes are known and well annotated. It's a big assumption. Sometimes you can read like databases like NCBI, RefSec, and UniProt KB. We usually take data from RefSec and UniProt. A big challenge again. What is reference sequence? It's just one representative sequence, right? If I start sequencing the genome of every student here, all of us here, do you think we'll get a 100% match even for a gene like glycerol, GAPDH or beta-actin, something that is so important? You will have single nucleotide polymorphisms that will change your proteome. Your beta-actin and my beta-actin might be very similar, but not exactly similar. And since it is not exactly similar, it might have something analogous to a RFLP in DNA. It might have that the trypsin cut sites have been altered. The peptides that your fragment will your proteome will generate and the peptides my fragment will generate are different. But the mass spec does not know that. Max quant does not know that. That becomes a challenge. Again, a big problem is the PTM or the post-translational modifications. Now, PTMs are invisible at the level of the genome. But what PTM does, it changes the mass. It changes the mass by changing of the mass, the M by Z data, the M by Z will also change. You agree? All of you agree this part, right? If I change my mass, the mass by charge ratio changes. So PTMs and genetic data cannot tell me where the PTM is probably there. So again, these modified peptides are thus absent from the database. So this again complicates the proteomic data analysis. So this is again one pitfall. So see, this session is all about pitfalls to make you sad. How do you do this? So incomplete. So another problem is this de novo sequencing and proteogenomics. I think we had discussed in brief what is proteogenomics in one of the lectures that I was giving. So incomplete peptide fragmentation and low mass accuracy. So as I said, HCDs or the fragmentation that happens at MS2 levels is again not always complete. It's sometimes a chance factor. The better your instrument, the better the fragmentation, but sometimes again the instruments are not working at 100% efficiency. The instrument efficiency alters. So when there is alteration in protein, these efficiencies, there is incomplete peptide fragmentation and low mass accuracy. There have been recent software improvements, like there is something known as the DIAN or the DIA neural network. So it is an AI-based tool that tries to correct for these changes. But again, there are limitations. What is AI? How do we develop an AI? These are models, and these models are developed on existing data. So last time Professor Graham Ball had addressed a webinar here, and he told that I would rather use augmented intelligence than use the term artificial intelligence. What AI does is make things easier for you, but it can never be creative. It cannot make anything new. It only regurgitates what information has been fed to it, and it will perhaps do a better paraphrasing of the information, right? But something like extremely novel, extremely new, highly unlikely, at least at this stage. So AI will also be limited by the same challenges that we discussed, that the RefSec database is primarily a RefSec. The entire idea, when we talk that we all humans are different, we have a different genome, and then I tell you, we have a reference sequence. Beta-actin sequence is this. GAPDH has this sequence. We are being hypocritical. How can we know a gap date sequence for sure if we are all different? It is very similar, but not the same. Now there is a solution to it, and the solution to it is you use proteogenomics approach. You sequence everybody's genome, and from that genome you do a 6-frame translation, and now you have all forms of protein in your database. But now you have made your database 6 times larger and your analysis sufficiently cumbersome and more processor intensive. So to solve one problem, you have created another problem. Another problem is amino acid alignment. Many peptides are not unique. Last time, I think in the Max Quant lecture, I would have shown there is something known as a unique peptide, right? A peptide that is unique to a specific protein, and then there are lots of peptides. They are not unique. Sometimes there is high similarity of amino acid sequence between species, and if there is a high similarity, you might put your input data as rat, and then you will have 4 human proteins suddenly coming up in your data. Shambhu would agree. You started off with rat, and suddenly your analysis is showing that you have 4 human proteins. The reason is they are so similar. Also, closely related species will have about 99% amino acid alignment, and distantly related species have 80% alignment. So what it does is, if you want to analyze your data, suppose you have a data set where you want to separate, right, you are trying to work with a host parasite relationship, and you want to check what are the host proteins, what are the parasite proteins. Now you know your proteome mix is actually both having host and parasite proteins. But fact of the matter is, sometimes you will identify a parasite protein just because it is very similar to the host protein. You might use 2 different runs on MaxQuant, so in which you will first use a human database and maybe then the parasite database. But since they are having similar, what will happen is you will get conflicting results. You will see there are lots of proteins which are actually not present in the host, but are actually present in the parasite. It will come up as a host protein and vice versa. Any impact on data analysis, also difficulties in identifying homologous proteins. Now there are proteins which are extremely homologous, especially that belong to families. GPCR family is one good example. So there we have lots of GPCR family proteins. Now when we want to do a mass spec on that, just because the peptides that they generate and the unique peptides are actually less, you will end up getting lots of data that is complicated by the very fact that there has been a misidentification because the proteins are so similar to each other. Another is in quantitative proteomics. So if you want to do a reliable quantitative analysis, you would have to use systems like SciLac, TMT, iTrack, and these instruments, these reagents are very costly. You can do label-free approaches. They're very cost effective. One sample run would cost you around 10 to 15,000. But if the MS conditions vary, if the LC conditions vary, again, analysis becomes a challenge. In label-free method, the biggest challenge is the technical replicate. The same sample you run thrice, you will get 3 different proteomes. There will be around 80 to 90% match, but even then, each cycle you will identify 10% new proteins. Now how do you go ahead? So you have identified 100 proteins every run, but out of that only 90% are common to all the 3 runs. Each run has identified 10 new proteins. Suppose you want to identify a biomarker. How do you go ahead? The only way ahead is you remove all those 10 unique proteins in each of the run and only go ahead with those 90 that has been repeatedly found. Something I think Shambhu would have demonstrated in MetaBioAnalyst, right? We remove, by statistical filtering, we remove all those proteins that are not in every run. Time, it's a big challenge. When you're doing a small projects, it's fine. If you're doing a big project, a multi-institutional project, and there you are trying to do samples in the tune of thousands. And as of now, in India, nobody does that, but we have European labs and American labs who do that. Label-free means each run will take 2 hours, then a 1 hour blank. Effectively, you can run only 8 samples per day. The reason those instruments run every time. Due to the different physiochemical properties, the peptide behavior will not be uniform each time. Electrospray ionization is good, but how they are ionized by this huge potential difference, that ionization is random. Every time the same peptide might not get ionized. Again a challenge. So it also, for all these reasons, it hinders the accurate quantification. Now since you think that these are the issues of label free, let's go for labeled, and all my issues will be solved. Sorry, we don't have solutions in this lecture, we only have lots of problems. Okay, the lecture might get demotivating, but I have 8 more slides to go. So if you go for any of these labeling methods, and I think I told you in the very beginning, no labeling chemistry is 100% efficient. Efficiency is low, and sometimes these labeling methods, like something like SILAC, is only applicable to cell cultures. So if you want to work on a live mammal, you have SILAM, but SILAM has its own challenges. There you need the rat or mice to eat isotope labeled food. If the rat is like me, it will eat more. But there are people who eat less. I will take up more labels despite being in the control group. And somebody who is a friend of mine who would eat less, they will take a much less label. Now when we are quantifying, there will be changes, right? So one more problem. So the next problem is false positives, false negatives, and unassigned spectra. So what is a false positive? Forget mass spec. What is a false positive?
Like the prediction is it's shown to be present, but it's actually not.
Right. So actually there is nothing, but you are shown that okay, you have this problem, or you have, so anything, like somebody tells you you are smart, but deep down you know you are not. So that's a false positive identification. But quite the opposite happens most of the time. Once you go for job interviews, you will get lots of false negatives where they will tell you you are absolutely dumb, you are not suited for the job, but you know you are suited for the job. That's a false negative. And unassigned spectra, as we had I think discussed in briefly, that there are just because it is not available in the database, but it is there in the actual data, that spectrum it's unassigned, right? Suppose I have a protein that is extremely novel, that makes me smart, handsome, intelligent, whatever. But that protein is only present. Suppose I am Superman or Batman, I can fly. Now I have that protein, but the proteome database at UniProt does not know that I have that protein that makes me fly at exactly sharp at midnight, right? So however costly may be your instrument, you won't be able to find that protein, because we are completely contingent on the database that we are using. So that spectra becomes unassigned, right? So persistent false negative identification, peptides and proteins may not be detected, and there are multiple reasons. Maybe the peptides cannot fly. Maybe the peptides by trypsin digestion ended up being less than 7 or more than 35. Can't detect. Physiological or pathological factors, lots of PTMs, modified peptides like phosphorylated peptides, acetylation, they will not match the database entry. You can't detect those as well. Rare PTMs like are often overlooked in searches. So if your PTM event is rare, it will again be overlooked. Unknown PTMs, there are a number of post-translational modifications that we do not know yet. And since we do not know, just like AI, it cannot tell us that what we do not know, right? So this is a big limitation. So what you can do is you can expand the searches to include more PTMs. Like if you go to MaxFont and set it up, you can include all known PTMs. But the moment you do that, time increases. Then you have to have a really fast computer, something like 128 GB RAM, and you have to go to a server. You cannot do it on a PC, right? Powerful software, extended analysis, of course, more electricity. Since you're running a server, now you need air conditioning, and you need 24/7 power. You cannot work in a system where there's a power cut, right? Because there's a power cut, the entire process stops, you again start from scratch. So all these big labs where they do lots of big data analysis, their servers are latched onto UPS systems that can serve till almost like 12 hours. So suppose there's a grid failure. The first day I entered my postdoc lab at IIT Bombay, there was a grid failure. Entire city of Bombay, which usually has extremely good power, the grid failed, so there was no power almost for like 20 hours. And everybody thought I would be a very bad influence on the lab. Artificial modifications during sample prep. So what can lead to false negatives? Methionine and tryptophan oxidation, cysteine oxidation, iodiacetamide modification, acrylbide modification of cysteine. So all these kinds of small changes can change your amino acid mass, effectively changing the ratio. And since we are always matching with the theoretical spectra, my theoretical spectra was supposed to be here, right? My spectra is here. Now this becomes unassigned. It's only when there will be a match, and perhaps this should be the perfect match. Even if it is matches there or there, the computer can make those corrections, right? The software can allow something known that we know as a mass tolerance. When we do some analysis, we allow a mass tolerance, that this much shift you can allow. But a mass tolerance of this, a wider mass tolerance, is not acceptable, because then there will be, all the results will be false positives. So the moment your spectra becomes unassigned, that becomes a problem, and these can be number of reasons why you can have these false negatives. Poor ionization efficiency, hydrophobic proteins and peptides. If your protein is hydrophobic, it is more often than not have a proclivity to not get ionized. It won't get ionized, and since it is not getting ionized, there will be a problem. Natural PTMs, glycosylations and disulfide bridges. So it will lead to mixed cleavage, something we know as the mixed cleavage. Mixed cleavage means there was a cleavage site. Since there was a sulfhydryl bond, cysteine bonding, double bonding, this cleavage site was missed by the enzyme. Now you allow for one missed cleavage, but sometimes if there are bad sample prep, you might have 2 to 3 missed cleavages. It can also hinder fragmentation and then lead to poor MS2 spectra. So MS spectra will be hindered because even the peptide, if it has to have a disulfide bridge, it will not allow the fragmentation. Of course, incomplete peptides, trypsin digestion. So miscleavages we have discussed. Incomplete, if it is a large peptide, more than 4 to 5 kDa, it will fragment poorly. We told it is much beyond the traditional range, and it will often be not identified without any specialized database. So suppose you are trying to look at a protein that only generates large fragments. You won't be able to detect. Jinal, is this helpful for you? Because you are the one that came up with the real problem in your research. Maybe you can go through these issues. Unassigned spectra, we just discussed. So approximately 60 to 70% of spectra remain unassigned. Can you imagine? That's why we have databases like PRIDE, where we put our spectra, and who knows, 5 years down the line, 10 years down the line, with advancements in computation, we can, and these 1,000 genome projects and 10,000 genome projects, we can know the human proteome much more better than what we know today. And maybe with better computing methodologies, we can again go back and reassess our spectra. So almost 60 to 70% of data is still lying in the database, not analyzed, just because spectra is usually not assigned to it. So what can we do? We can make customized database searches. You can customize your database. And of course, we need significant advancements in bioinformatics as well as databases. So these large genome projects that we are having, we can have, for each allele, we can have a different spectra. And then maybe we can just look for a specific allele. We don't want to look for the entire proteome. We just want to look for a specific set of alleles. So I think his battery ended, and it was a signal that I should end my talk. So I would end my talk with a very nice paper. I think we will share it with you all. Like as I said, today evening when you go back home, or maybe tomorrow morning, you would find a drive link. So there are 2 things to do with the drive link. One is try to read all the papers that we share. And number 2, I will ask all of you, you must have taken a lot of photographs. Do share the photographs if they are share worthy, and specifically share if they are not share worthy, so that we can have a meme fest in the group sometime later, right? So that's all. I think that's my final lecture for this part of the day. I will now move for some admin work, and always remember, don't stop questioning. That's, I think, in today's date, when we rely so much on ChatGPT for almost everything, don't stop asking questions, because I think that's the most important thing, that's the most beautiful thing that human does. Whatever we have achieved is because of our curiosity, right? We are the only species who do that, because we have a very highly developed prefrontal cortex, makes us do things which a dog would find extremely unnecessary, right? A dog does not have a planned meal. A street dog would roam around. Whenever he finds something, he'll eat. If he doesn't find something, he won't eat. He will just go and sleep. You can't do that. We have long plans, right? All of you are here with the plan that, okay, I will learn something, then I will perhaps apply for a postdoc, perhaps I will use this in my research, perhaps when I am passing out my PG, I will apply to a proteomics lab, so it will help me. So we have long-term plans. Dogs don't do that. Again, all because of the prefrontal cortex. So please be curious enough. Don't stop questioning. And also, this is a hint that you can ask questions if you have any. Else, the tea has been waiting for you. You can join us for tea. And did I bore you? I mean, did I break your heart today? It was a planned exercise. I think you already knew from the list that on day 5, we will know where proteomics fails. But I think after you learn that proteomics can do everything, you need to realize, just like any technique, it has its own pitfalls. And it's very important when we plan our experiments not to get over enthusiastic and try to plan an experiment that is more pragmatic, that will lead to good results. Despite these issues, we have done considerable advancements in proteomics because of proper planning of experiments. Okay, so I think I will leave here. Okay, you have a question.
Sir, what would be your plan to delve into the proteomics of a species which is not very much known in that field, like which hasn't been discovered in the field of proteomics much?
You want to work on a species that is that nobody has worked on?
No, you have some papers, like 2-3 people have worked on this proteomics, but it is not in that directed way which I am working on. But I want to delve into the proteomics of that species, right? Because in psychology, we identify new species every day.
Right.
So
has anybody done, like, if you don't go for the species, did anybody work on that specific class or subclass of? So you don't have to work on that specific algae. What was the work done on algal proteomics? Right. I would start from there. If I have to work on a fungi, I will look what is the status of fungal proteomics. What are the databases they use? So there are a lot of things that you can use, like there are multiple softwares and that people use, which are actually helpful to try to extract data. But as I said, even with a known proteome, you just saw what are the challenges. There is almost 50 to 60% of unassigned spectra. You would lose out a lot of information. You can go for targeted approaches. Maybe you can, if you know specific peptides, you can create a list that, okay, you want to look at only 50 or 60 proteins, and you can run your experiment on a triple quad. Rather than doing a discovery approach, you can go for a targeted approach. You can only go for something that is already known, whose parameters are known, whose sequences are known. Because sometimes going for discovery at the level of proteome is difficult. Or you can go for the proteogenomics approach, where you can first start by sequencing the genome of your species, right? And do a 6-frame translation and create your own database. And then you do discovery data. Of course, working on non-standard models is difficult. So your thesis can be just establishing a proteomics pipeline to work on your specific species of algae. That itself will be a 2 to 3 year work, sufficient for a PhD. But now maybe somebody from your lab can take that pipeline that you have developed. And of course, when I'm talking of a pipeline, I'm talking of a lot of things. Like when you go for a 6-frame translation, you see the number of proteins that you identify will incrementally go up. Then you have to validate those proteins using targeted methods. So I think I won't step into that mine if I had to do more work with it. Only proteomics, that itself is 4 to 5 years. I mean, 3 years is a very good if it is something that you have a mass spec at your door that you can access daily. But even if it's a central facility, you would be fighting with 10 other labs for the same facility. And again, challenges when there are central facilities. Now they would run some sample, right? And few peptides stick. They are very sticky peptides. Once they run their sample, they have effectively compromised the column. So it happened in our lab as well. So even in the lab, there are multiple people working, right? Multiple groups working. So there was a specific contaminant that was coming up in one of our data sets. And we did not know why it came in some and it did not come in others. So what happens in a lab is we randomize the samples, right? We randomize the samples so that there is no bias. Because if you don't randomize, you will be effectively biasing your experimental data. So what we observed was this particular, actually 2 to 3 protein set would always come when person X's sample was lined up just before ours. But it took 3 to 4 months to realize that. And by that time, we had already gone like 30 or 40 runs. So what we did was we excluded it in our analysis. But again, that could have been avoided. So these things come up when you practically start doing stuff, start preparing samples. Lots of things. I think I have tried to cover almost all bases. And the review article that you showed you is a very good article. It also shows the strengths and the weaknesses. I focused on the weaknesses because that was the theme of today's talk. Anyone else? Yeah.
So can we regenerate the nerve cell that has been degenerated through proteomics?
Yeah, you can do discovery proteomics. But you cannot regenerate per se, but you can understand a developing nerve cell. So nerve cells develop, right, from stem cells. So a stem cell that is differentiating into a nerve cell, you can try to understand its proteome dynamics, right? Now you try to see what are the events that are leading to its full generation. Fine. Now you take a degenerating nerve, understand its proteome dynamics, and then you can do a systems level analysis and try to see what are the proteins that are getting turned on while it is generating and when it is degenerating. And based on that, you can build up a hypothesis that if you can turn on these specific proteins or these specific pathways, then you can think of something like a regenerating a neuron that is already dead. So you have to go back in that way.
So like whenever we are to find targets or anything, so we just go for say if in proteomics we go for a protein, but of a single protein we have different isoforms also.
Right.
Like in some studies it has been reported that this isoform is significant in this target.
Yeah, I mean phosphorylations is for example.
Usually in normal cases we just go for the main form of protein. We don't look for isoforms. So like how we should deal with it if we are into discovery proteomics? So should we go for like normal protein kind of thing, or how we should look for isoforms?
So you see, you cannot randomly look for an isoform, and because I just told you what are the challenges of randomly looking at something. One thing you can do is develop an hypothesis. Suppose you are working on a system that has lots of kinases, right? You're working on cell division, and we know there are cell division cycle kinases, right? So, which means there will be a lot of phosphorylation events. And since there are lots of phosphorylation events, you can try to design a phosphoproteomics pipeline. Phosphoproteomics. So we did not go into deeper into that because perhaps that would be a different Gyan course, where for 5 days we will just talk about specialized, extremely specialized proteomics approaches, but then the target audience will become extremely specific to those who do proteomics at the level of PTMs. So if you do phosphoproteomics, then phosphoproteomics becomes a big challenge, because I mean, it's not a big challenge. Then you have to start preparing your samples likewise. I mean, it starts at sample prep. Most of the things you would understand starts at sample prep, because sample prep will significantly alter the kind of output you will get. So yes, randomly you can do that, but I'm not very sure about the reliability of the data. But you can set on max quant, there are parameters where you can set to look for changes in the phosphoproteome or acetylations, but they are not very accurate, because most of the changes that you would be seeing might also be changes that you induced while processing the peptides. I think I can take one last question, because then we are getting late. Professor Graham is waiting. Andromeda.
Yeah, in order to create the theoretical spectra, we use input protein sequences, right?
Right. Faster sequences.
So where do we get the sequence? Is it because like we do not know the entire protein profile of whatever species we are working in. So in that case, if at all we are giving some input sequence.
So Andromeda, the FASTA file that you can give can be anything, right? The FASTA input can be, you can take, suppose you want to expand your search. So rather than going for UniProt, you can go for Tremble, right? A translated EMBL. So that will significantly increase your hits. So the number of identified proteins will go up. But since we do not know whether these proteins actually exist, it becomes a challenge. You can sequence your own genome. You can do a whole exome sequencing, and from that genome you can do a 6-frame translation, use a proteogenomics approach, and make your own database. But every time you do that, the larger your FASTA file that you upload onto Andromeda, the larger the number of targeted theoretical spectra increases, and now the instrument will try to match first at the level of MS, then at the level of MSMS, and then give you the data. So theoretically, yes, but again, availability of time, availability of resources, all those will determine what best could you do. So you have to, what's your name?
Kishore.
Kishore. You are from?
Manipal.
Manipal. The Manipal, I think, has a mass spec. Which one is that? Right. I guess they have a fusion. So most of the central facilities that we have, they run thermo instruments. I think that way Professor Sanjeeva's lab is one of the first in India to start with the Bruker timsTOF. So maybe in the next 4-5 years, now since Bruker has set foot in India, you would find more and more timsTOF. So there are multiple things that you can do with mass spec. There is something very interesting, that is mass spectrometry imaging. There you don't take samples in a solubilized form. You take a tissue section, right? Now imagine something as complicated as a brain, right? Number of cells, the way the cells are arranged, you have the hippocampal formation, if you have the prefrontal cortex, you have the cerebellum. Now if I homogenize them all, I'm not getting the exact data, right? I'm getting a projection of the data, what it might look like, but not the exact data. So there you can do mass spec imaging. Mass spec imaging will give you this chemical map of which protein is present in which area of the brain. Also, there are other methods like thermal proteomics, chemiproteomics, so lots of things to do. I mean, you can explore a lot. I think this Gyan course is primarily to act as a primer. So we have added the PCR primers. Now if you have enough of Taq polymerase and you are ready to go through those rigorous thermal cycles of heat and cold, then you would have a long amplicon. Okay, thank you. So please have your tea, and if you have not taken your attendance, do